Okay, good afternoon. It's my pleasure to introduce our speaker today, Dr. Jingyi Jessica Lee, Professor of Statistics and Data Science from UCLA. Jessica's research lies at the interface of statistics and biology, with a focus on developing statistical methods to understand large-scale genomic and transcriptomics data. Jessica earned her bachelor's degree in biological sciences at the Tsinghua University and PhD in biostatistics from UC Berkeley. She joined UCLA in 2013 as a faculty. Since then, she has made significant contributions to computational biology, especially in creating tools for single-cell RNA sequencing analysis. She's a leader in guarding rigor and transparency in tool development, a strong advocate for using synthetic data to benchmark computational algorithms, and a creative, tireless scientist that we come to know and appreciate. She collaborates broadly to pursue topics such as classification problems and multi-species joint analysis. Her record of innovation has earned her numerous prestigious awards, other than the ones you know, such as NSF Career, NIHR 35, and those from Sloan, Keck, or CZI. She has also received, just bear with me here, received the Overton Prize from the International Society for Computational Biology, the Emerging Leader Award from the Committee of Presidents of Statistical Societies, MIT Technology Review's Award for 35 Innovators Under 35, Johnson & Johnson Women in STEM Scholar Award, and I've got a chance to hear her Radcliffe Fellowship from Harvard that gave her a whole year of visiting professorship to connect with scholars from an exceptionally wide range of disciplines in natural sciences and humanities. So it's, again, my pleasure. And I want to thank the seminar committee led by Minji Kim and the Kim Fai-O for inviting Jessica. Now, please join me in welcoming Jessica Lee.
Thank you so much, Professor Jingyi, for the very generous introduction. And also thanks to Kim Fai and Minji for inviting me to give this talk and visit this very vibrant department. And it's my great pleasure to talk about some of our recent work just from the perspective of permutation. It's a new talk. Actually, I reorganized my old stuff into this new talk to provide, I will say, hopefully a simple but useful perspective, and which I hope everyone can integrate into your method development or data analysis. So just as Professor Lee introduced, my research has been, we try to sit at a junction of statistics and biology. So that's how I designed the logo. If you pay attention to it, JSB, and the two colors, one for statistics, one for biology. So I also use the same two colors to highlight the two concepts from my title, permutation and genomics. So just to gauge your interest from the beginning, yesterday you might have seen the big news for the Nobel Prize in Physics. And I noticed this very interesting figure in Chinese news media. And I kindly asked my postdoc Han to help me translate this to English. So basically, if you don't look at everything, right, it's just two dimensions. One is the object we study. So we can go from fundamentalism to liberalism. So it can be very broad or very specific. And we can also have the method going from fundamental to liberal. So what it means is that we will have less requirement in defining what is physics. So this is where we are. Neural networks is also physics. But this liberalism made me wonder the connection to our research, right? If we think about genomics, I would call it a liberal discipline in 4 ways. First of all, it's interdisciplinary in nature. Second, it has a very data-driven focus instead of theory-driven or hypothesis-driven. If it's data-driven, you may end up getting many interesting findings from data. Third, we have a very rapid evolution of methods, as you can see, which I will show very soon for single-cell data analysis. Fourth, it has allowed flexible analytical approaches. So we have them, if you have real data experience, you will know how many steps are involved in your data analysis, and different people may do data analysis in a different way. So this can be seen in this website, the single-cell RNA, scRNA tools.org. So you can see the quick evolution, right, of methods. We ended up getting more than 1,000 or 1,500 methods for just single-cell data analysis. So facing this success, huge success in AI, right? Nobel Prize in Physics even, and also this data analysis liberalism in our field. So the very fundamental question, I would say this is a question I had even when I was a PhD student at Berkeley. So is statistics still relevant? Because statistics itself is such an old discipline, more than 100 years old. Given we have so many tools, so many flexible approaches, and we're being so liberal in doing genomics, is it still relevant? In my opinion, statistics is a discipline that ensures rigor in data analysis. So to me, as long as we care about rigor, it still matters. So why do I say this? Let's go back to the history, to the origin. So one of the most famous and fundamental method you guys learn from Intro to Stat course is the two-sample t-test. I happened to have the opportunity to visit the origin of this method is actually in the Guinness Brewery in Dublin, in Ireland. So this guy, whose name is Gossett, he actually worked at this brewery, and he was interested in comparing the quality of their beer in two batches to see whether the new technique improved the overall quality. So to answer this question, he needed to compare two sets of numbers from each batch. So because he was so humble, he named himself using the pseudonym of student. That's why it's called student's t-test. But basically, what this is about is that we want to compare the two averages or two means of the two sets of numbers. So even without statistics, we could do the comparison, right, as an elementary school student. And there should always be some non-zero difference. But the question is the difference big enough to say, yes, we have a better quality with a new technique? This is the question statistics help to answer. So you can see even back then, it's about the rigor in data analysis. How do we interpret data? How do we make conclusions? Continue from here, I really like this article and I strongly recommend it to people here. It's about this summary of important statistical ideas in the past 50 years. And Andrew Gilbert is a renowned statistician at Columbia. And there's an even, how to say, simpler summary on Medium about this article. So I will draw my talk from a connection in this article because it mentioned, I think it listed 8 important ideas. And the second of it is called bootstrapping and simulation-based inference. Under this idea, there's a quote. In permutation testing, resample data sets are generated by breaking possible dependency between the predictors and target by randomly shuffling the target values. So permutation is the theme of my talk, and I will show you 3 examples on how it can help with my research. So to begin, I will first show you 2 scenarios about permutation. So briefly, right, in many machine learning problems, data analysis problems, we have a supervised learning setting in which you have a matrix. And here I'm just denoting the rows as samples, columns as features, and you have a company response or outcome vector called Y. That's called supervised setting. And for unsupervised learning, you just have this matrix X, no Y. And in my talk, I'll give you some respective examples. For bulk RNA-seq data, which I'll talk about, it's actually under this setting. So here the features are genes. Samples are just biological samples. And Y are some sample condition labels, binary. For the unsupervised learning setting, my single cell examples will be under this setting. Here the samples are cells and the features are genes. So the 3 examples I will give you, the first one is for bulk differential expression analysis, second for single cell data visualization, and the third, the most recent ongoing one, is the aggregation of single cells into metacells. So the first 2 works were published. So this bulk RNA-seq DE analysis is one of the most commonly performed in the analysis in our RNA-seq data analysis, I should say. So differential expression is defined as, if a gene has different expression levels between two conditions, I call the gene a DE gene, differential expressed gene. So this cartoon shows a typical scenario in which you have collected experimental replicates under each condition, because RNA-seq in those days was quite expensive. So people typically collected 3 replicates per condition. And this comparison is done gene by gene. You look at one gene at a time. So intuitively, we want to call the left gene, which shows greater differences between the two conditions, a DE gene. The right gene, maybe not so much. So statistically, it was done as a hypothesis testing problem, one statistical test per gene, with a null hypothesis that the gene has equal expression under the two conditions. So if we decide to reject the null hypothesis, we call the gene a DE gene. That's the setup. For this analysis, if you want to learn how to do it, the 2 popular methods you will definitely encounter are HR and DCMB2. They were both developed more than 10 years ago, and they were designed for small sample sizes, like the 3 versus 3 scenario. So it's a small sample size, and we know that it will be a big challenge to get some reasonable statistical power because your information is so little. So therefore, they have to compensate the small sample size by assuming a distribution of data. RNA-seq data are counts. If you summarize the data, right? One gene get 1 count in 1 sample. So they assume, both methods assume this negative binomial distribution for count data, which is more general than the Poisson distribution because you get an additional variance parameter, which could be bigger than the Poisson mean. If it's Poisson, you just have 1 parameter. And furthermore, both methods will account for the fact that different RNA-seq samples have different sequencing depth and sample library sizes. So in this formulation, essentially, we assume 1 negative binomial per condition, and we assume that the condition has its mean parameter mu1 and mu2 multiplied by the sample-specific size factor. So given that, then you have some independence assumption for the counts of the samples under each condition. So for these methods, both methods, the null hypothesis is that mu1 is equal to mu2. That is subject to sample size differences or size factors. We believe that the 2 negative binomials should share the same mean parameter. I would say this is a reasonable hypothesis only if the negative binomial assumption holds, because it's what you assume for the data. So our surprising discovery was by coincidence in a collaboration project with Professor Wei Li at UC Irvine with his former postdoc, Yu Mei, who is now a faculty herself in China, and my former student, Xin Zhou, who is now a faculty himself at Oregon State. So this collaboration started from the journey where Yu Mei applied DCMB2 and HR to this data set, which is about immunotherapy of patients' blood samples before therapy, which is 51 patients and 58 patients on therapy. And it was published in the Cell paper, the blood RNA-seq samples from patients. So her first finding was that DCMB2 and HR actually give quite different numbers of DE genes at the same false discovery threshold, say 5%. So HR found almost twice as many as DCMB2. And their overlap is not so big. So the intersection is not so big. So this motivates Yu Mei to look into this question and ask this question: why do DCMB2 and HR identify drastically different DE genes? Because here the sample sizes are 51 and 58. Then I had this question. So in these cases, can we do some permutation? Because there are many ways we can permute the data. So just imagine that if you just want to randomly assign a total of 109 samples into 2 groups of 51 and 58, you can do 109 choose 51, right? That can be a large number of permutations. So let's just try that. And I call this condition label permutation, because once we permute the labels, there's no more relationship between the condition label and the gene expression. Then we get a more surprising result. That is, if you look at here, so from the permuted data, each permutation, we can apply a method and get a DE gene number. And we can do many, many permutations and then get a distribution of DE gene numbers. whose average is the height here and whose standard deviation is half of the error bar here. And you can see that from many permitted data, we get even more DE genes than we get from the original data, which is more surprising. And how could that be possible? The reason is the negative binomial assumption does not hold for this data set. So just as I mentioned, the assumption was first assumed for replicate data, right? Experimental replicate data under a control condition. So we can have some assumption about the sampling variance due to Poisson, and then we can assume that maybe there's some biological variance in the 3 replicates, subtle variance, and then Poisson gave us the counts. And together, like for example, gamma for biological variance and Poisson for sampling variance, together we get a negative binomial. But these samples are from patients, and patients could be more heterogeneous than they should be than experimental replicates. So that's why our first check is to use the 51 data points and 58 data points to check whether the negative binomial fit well to data. And these are based on the edge-R's fitted negative binomial. So these are called quantile QQ plot. So if the model fits well, we should expect to have some, like the dots should lie on the straight line, but that's not the case. We have some clear outliers. That's for edge-R. And for DCMB2, another gene, we also see the similar pattern. So basically what's indicated here is that the negative binomial assumption is not reasonable for this data from human patients, not replicates. And furthermore, we divided the genes into 2 groups, 2 extreme groups. One group includes the genes that are rarely identified from permitted data, the OK genes. And the second group contains the genes that are frequently identified from permitted data, the bad or problematic genes. So between the 2 groups, we check whether the fitness or goodness of fit for negative binomial for genes in each group have some differences. And this shows that, okay, let me just explain. The y-axis is the negative log p-value for goodness of fit test. The smaller the p-value, the worse the fitting. And because we take negative log, we have larger value to indicate worse fitting. So in this case, it confirms our guess. That is, for the problematic group, negative binomial fitting was poorer. And I want to give a credit to my current master student who did her undergrad research in Chinese University of Hong Kong. After she read our paper published, she pointed out an issue in our goodness of fit p-value calculation, and we fixed that. So the one I'm presenting here is the post-correction figure. The good thing is that our conclusion qualitatively still holds, but we have to fix the scale here to make it more accurate. So coming from this conclusion, like negative binomial assumption doesn't hold for this data set, then the next try would be for us to use a test that doesn't assume negative binomial assumption. And we use the Wilcoxon rank sum test, which is a so-called non-parametric test. It has a different null hypothesis. Basically, I'm making it simple by assuming my data are normalized, so I don't need to worry about the sample size differences. With a normalized count, the null hypothesis of Wilcoxon rank sum test is that if you just randomly pick one value, one count, normalized count from condition 1, and you random pick another one from condition 2, then which of the 2 numbers is bigger is equally likely. So you have 0.5% chance that one is bigger than the other. So it's based on the relative ranking of values instead of saying negative binomial and the same mean. So this doesn't have the negative binomial assumption. On this data set we look at, even though the result may be disappointing because Wilcoxon didn't give us any discoveries. However, it's not self-contradictory because it also didn't produce anything from permitted data. So it's consistent. So I just want to say that this is not a benchmark study because we didn't include all possible methods. I know there's maybe like 2,000 methods out there. It's not a benchmark study. We just want to say that the assumption matters. And if we just use popular methods without knowing their assumptions, we may get false discoveries. So surprisingly, this this analysis paper got a lot of attention, especially on internet after publication. And some people even tell me, I mean, someone from UCLA Medical School told me, oh, this is your most famous work. And I was very shocked because it's not a new method, it's analysis. But what it told me was that, wow, something we computational people took for granted, like we need to check a method's assumption, is not as obvious to people who analyze data. So that's why I feel like permutation is something people can implement in their practice. But the discussion continues, I would say. So this is like, how to say, I shouldn't complain, but basically our correspondence should have been published half a year ago at Genome Biology, but we're still waiting. So this correspondence is actually, well, we're responding to two correspondences that pointed out either an issue in our analysis or a suggestion. So one correspondence saying that we neglected normalization in our analysis or in our simulation part. So we definitely, so I should say, I shouldn't go to too much detail, but we did some correction to our analysis and said that we want to argue that if the semi-synthetic data based on simulation does not assume any batch effects in its definition of true DE genes, then no normalization should be applied. So this is something we should also pay attention to because real data analysis involves normalization, but our simulations based on our assumed ground truth. So in that case, we have to be careful about whether normalization should or should not be used in our simulation. Second correspondence pointed out that if we apply a procedure called Winsorization, it's just a fancy way to say outlier removal. Basically, you remove the outliers from your data, and then you can run DCMB to an HR. So this correspondence shows that without the outliers, the false discoveries we reported were not severe. However, we want to argue that even though this is true, but in real data analysis, throwing away outlier is not as straightforward as it is, right? Because you need to set a threshold, how much to throw away. So basically in this correspondence, we just showed that Wilcoxon remains to be robust, even though we consider normalization or Winsorization. But I just want to say that there are many choices in real data analysis, and whether you want to go with a robust method or whether you want to go with a delicate method specific for this data set, you need to make this choice. That's the first example. Second example, single cell visualization. So this is an example where I will show under the unsupervised learning setting. We just have the matrix X. So the motivation is about the common use of t-SNE and UMAP for visualizing single cell data. This is a very well-written blog for t-SNE. So in short, when you use it, you need to pay attention to the hyperparameter because both methods are nonlinear dimension reduction method. For t-SNE, there's an important parameter called perplexity. Perplexity roughly measures the neighborhood size. So basically, if you set the perplexity to be small, you're assuming every cell's very close neighbors are neighbors, and you want to preserve the neighborhood. When you make the perplexity very big, then you have a very large neighborhood size. So both t-SNE and UMAP are good at emphasizing clusters. So you can see clear clusters. And this is a simulation example in which the real data, the truth has just 2 dimensions. So this is the true visualization. But you can see that when you vary perplexity, sometimes you're closer to the truth, sometimes you are very far away from the truth. This issue was also reported recently by a Nature Methods journalist saying, how do we see data as t-SNE and UMAP? So basically, there's one sentence in the abstract. Sometimes these methods take a second thought. We have to use them carefully. So the question, my collaborator, Professor Lucy Xia, who is a statistician in Hong Kong, and my student, Christy Lee, the problem we try to tackle is to answer this question. Can we tell whether a cell's embedding is dubious or trustworthy in a t-SNE or UMAP plot? Our idea is to decide on this trustworthiness by looking at the cell's neighbors before we run t-SNE or UMAP and after we run t-SNE and UMAP. This is our intuition. So basically, we look at 2 sets of neighbors. And this cell 1 is one good example I want to show using this cartoon. That is, before we do the embedding, we can define some neighbors for the cell 1.
After we do the t-SNE embedding, we can define the neighbors. So on both sides of this slide, I'm showing the same t-SNE embedding or UMAP embedding, doesn't matter, it's an illustration. Same embedding, but I'm labeling two sets of neighbors, one based on the pre-embedding space, one based on the after-embedding space. So to just clarify, because we hope to make some reliable conclusion about the relative locations of cell clusters to each other in this 2D plot, we set the neighborhood size to be half of all cells because we want to capture or check the preservation of mid-range distances. Furthermore, for the pre-embedding neighbors, we actually defined it in the principal component space because that's the standard practice in the single cell field. That is, you first do some log transformation, then PCA on your data, then you keep about 20 to 50 PCs, a number of your choice. You then input those PCs as the input into t-SNE UMAP. So in other words, you're not using t-SNE UMAP to reduce the original thousands of dimensions as genes, but the PCs. And also, in both t-SNE and UMAP, they operate on the Euclidean distances of cells in the input space, the PC space. So therefore, when we define pre-embedding neighbors based on Euclidean distances in the PC space, and we define 2D embedding neighbors based on Euclidean distance in the 2D space. So then what do? We want to check the consistency of two sets of neighbors. By consistency, we mean that whether the ordering of neighbors are consistent, and also whether the distances between the neighbors to the cell 1 in the 2D space is consistent. So here, we just order these neighbors' Euclidean distances in the 2D space to cell 1 by following their order in the respective space. The reason is that we think when we look at this plot, we are visualizing the 2D Euclidean distances. So that's why that's what matters. And then with these two ordered Euclidean distance vectors in the 2D space, we check their consistency. And we use the Pearson's correlation to measure the consistency because we need to care about the absolute values, the magnitude of distances, not just their ranks. So this is a good, trustworthy cell embedding example. This is a bad example. I just want to show that the cell's neighbors change a lot from pre-embedding space to the post-embedding space. Okay, so that's the intuition, but we need to formalize this intuition. And our question is, what is the null hypothesis in this problem? So I would say it is that a cell's neighbors, half of all cells are just random after embedding. So before and after embedding, you have a random selection for half of the cells as neighbors. But how do we obtain such a null hypothesis? How do we realize that? We can use permutation. So in this case, I'm just thinking about one case in which I can permute every gene independently. Okay, so then the cells became permuted cells because they're no longer the real cells. But every gene still has its marginal distribution preserved because I'm just shuffling the numbers for each gene. So I do preserve every gene's marginal distribution, but I didn't preserve gene-gene correlations or cell-cell relationships. Since I do the independent permutation for every gene, essentially I get a new population of these permuted cells. And in this population, the permuted cells have exchangeable relationships. They're equally likely to be close to each other. So that gave me the null hypothesis. And then we implement that. So the permuted data is just the permuted cells I talk about. And I just need to do this permutation for once for my whole data set because every cell becomes a permuted cell. And then I can calculate my score for every permuted cell. And these scores together give me a null distribution. So this reflects what my score would behave if there's no real relationship among my cells. So under using this null distribution, I can calibrate my score for the real cells, whether I believe the score is high enough so I can call this real cells embedding trustworthy. It must be much higher than the null scores. Or if the score for a real cell is so bad, that is even worse than most of the null scores, I call that dubious. Just one message is that here the percentages of dubious and trustworthy embeddings are not just the two tails because I'm using the null distribution, not the real cells distribution. So in fact, on a real cell population or real cell data set, we found that majority of the real cells can be called trustworthy, which is good. And minority is dubious, and we don't have so much in the middle, the gray zone. So looking at one example in our paper, and we have multiple examples, but this is the most striking one. This is the first Hydra single-cell RNA-seq data set published in Science in 2019. So this is the original visualization. We were able to reproduce the visualization. And we run our algorithm to detect dubious embeddings, and they are labeled as red. So you see what's interesting here is that many dubious embeddings are those small clusters. And if you don't know that, if you just look at the t-SNE, you might say, oh, these small clusters might be cell types or cell subtypes. But if you know that their locations are dubious relative to other cells, then we should be careful when we draw conclusions. This is one use of scDEED, the method, to detect dubious embeddings given one perplexity, given one parameter. And another use is that we can change the perplexity parameter, and then for each parameter, we detect dubious embeddings and count the number. So we can minimize the number of dubious embeddings and as a way to optimize the hyperparameter perplexity. So for this data set, we ended up getting the perplexity to be 230 instead of 40. So you see, comparing this new visualization to the previous one, we see some big differences. In particular, if we focus on these three colored cell clusters, previously the blue and purple one, green and blue ones were like surrounding the orange one. The orange one is a big cluster of stem cells. So you see that these neuronal ectoderm cells were surrounding the orange one in similar distances. But after our visualization optimization, these three clusters are unified down here, very far away from the orange one. So if you look at the data, the gene expression data in heat map, you can see that our visualization is more consistent with the data. So these small clusters are indeed not so different, and they're very different from the orange cluster. So that's my example 2, showing that when you use permutation, you may help you decide whether visualization is reasonable or not. The third example is the recent work, which hasn't been published yet, but we hope to post a preprint very soon. So this is called the metacell concept, which is very interesting because you change your data. You no longer work with single cells, but you aggregate them into metacells for the purpose of having less sparse data. So in this cartoon, if you just have the single cell matrix, this is from this paper, and you basically can, so basically you have the cells as the columns, genes as the rows. This is different from my original X matrix because I regard cells as samples. But anyway, if you aggregate certain cells into one metacell, you get a smaller matrix. So here your columns are metacells, and you get fewer zeros because of the aggregation, from 90% of zeros to 50% of zeros. But from a statistical perspective, this is interesting because the question is there a reasonable definition for aggregating cells into metacells? Obviously, we cannot aggregate too much, right? If you aggregate everything into one metacell, there's no information. Or if you don't aggregate, you should just work with single cell data. So there must be some trade-off there. And this is the literature review. So far, there are 4 metacell methods published. From the first one, metacell, metacell 2, they were from the same authors. There's one called supercell, and the most recent one is from Dana Pierce's group called C cells on Nature Biotechnology. So they all use the metacell concept and aggregating cells into metacells in different ways. We also have many prominent application papers. They are all Nature papers, right? So basically, these papers implement the metacell concept, even though they may or may not use the methods here. So it has been used in practice. But if we read those papers, there's no consensus on metacell definition. So this motivated us to think about this problem. Can we define a metacell in a rigorous way? And this is the first metacell paper. I'm just mentioning the quote here. It defines a metacell as a homogeneous collection of single cell profiles that could have been resampled from the same original cell. So essentially, we translate the sentence to say that we think the variation of cells within a metacell should be attributed exclusively to measurement or technical error, not biological differences. And this concept or this question motivated me to look into, or actually I read this paper before, so I draw the connection here. It was from Matthew Stephens' group. It's a very interesting paper, very well-written paper on Nature Genetics about separating measurement and expression models in single-cell RNA-seq data. So this paper was originally about the zero problem or imputation problem, but it's very conceptual. It's related to this metacell concept. So in this paper, the authors proposed two-layer models. One for expression model, that's the distribution of the true expression levels of genes in cells. And the measurement model on top of that, that is the distribution of the observed counts given the true expression levels. So this is probably the most technical slide in my talk, which is how to describe this two-layer model. So again, we consider a cell to be an observation from one distribution, cell I from 1 to n cells. We consider a gene to be a feature, J from 1 to P. So this two-layer observation model has the first layer about the biological true expression. So here I denote this to be lambda I for cell I. It is a multi-dimensional vector because we have all genes here and p-dimensional vector. So the expression model describes how the cell's true expression levels for the p genes are distributed. So in particular, there's something to pay attention to. Lambda ij is defined as the relative expression level of feature j, like gene j in cell I. So it's the proportion. And all the lambda ij's across j sum up to 1. Okay, so it's the proportion. Why do we care about proportion? Because sequencing data only give us the relative proportion. We don't have the absolute abundance level. So that's why it's more reasonable to talk about lambda ij than the absolute number of features in cell I because we don't observe this. And this F is the distribution model, which is p-dimensional. Then given the lambda ij, the measurement model is used to describe how our observed count is distributed. So Yij is the count of feature J in cell I, and Yi plus denotes the library size of cell I, the sum of all counts. So basically, the measurement model is like, given the true proportions of features, given the cell library size, how would you allocate the total into individual features? And this is a one-dimensional distribution just for the measurement. So together, if we want to have a statistical definition for a metacell, we want to say that it should be a group of single cells that share the same lambda, because lambda is the biological signal. Okay, so we just want to have an approach to check this definition. If we think a metacell satisfies this definition, we want to call it a trustworthy metacell. Otherwise, we want to call it a dubious metacell. So we're borrowing the names from the previous project, and we want to formulate this as a statistical problem. So why would dubious metacells matter? So this is again from this review paper for metacell at MSB, Molecular Systems Biology. You can check out this paper. So it has this nice cartoon showing that if your metacell happens to include cells of two different types here, then it will bias your gene-gene correlation analysis, because this is what you get using single cells. This is what you get using metacells. And if you have a dubious metacell here, you will get a different correlation calculation. So how do we tackle this problem to check whether some metacells are dubious, some are trustworthy? How can we screen out the trustworthy ones? So my postdoc Pan Liu led this project, and our first question is, can we achieve these goals? So we hope to give you a statistical criterion based on which we can identify dubious metacells consisting of single cells from different cell states. These are the problem ones we shouldn't use. And we also hope to nominate the top performing metacell method out of the 4 and optimize hyperparameter. So every method has a hyperparameter which can be translated into this granularity level. That is, on average, how many single cells do you aggregate into a metacell? We hope to choose this in a data-specific way. So our intuition here is that within a trustworthy metacell, features are approximately uncorrelated. This is the most important part because we think the differences of these single cells in this metacell are just due to the measurement error. And the measurement error should be roughly independent for different features. So they should just give us a weak correlation. And I have to say it's not zero correlation, but weak correlation. The reason is that because we have the cell library size as a total, so that will still induce some weak correlation, but that's pretty much it. So this is from like a real data. We can show that for a trustworthy cell we found, metacell we found, the gene-gene correlations does not show a block structure, but from a dubious metacell, we do see some correlation structure. And our strategy is to propose a per metacell statistic. We call it MCDIV, standing for metacell divergence. And also, we hope to get some reasonable node distribution by permutation. So this is the third permutation I will introduce today. First question, previously for scDEED, I used a within gene permutation. Is it enough here? So we permute every gene independently, right, for that purpose. So we can get the permuted cells with exchangeable relationships, no real relationships. But here, the problem is that the cell library sizes are disrupted because once you permute every gene independently, each permuted cell doesn't carry the original cell's library size. But in the metacell definition for the measurement model, if you remember, it involves the cell library size because we're working with counts, not normalized counts. So the cell library size should be preserved. So then this is an interesting strategy Han came up with. I call it double permutation, which is like for this top row, we permute every gene independently just as a way to serve as the, negative case where genes are uncorrelated. So based on the original matrix and this permuted matrix, permuted gene matrix, we can get our statistic mcDIV. And I ignore the detail here because our work is still ongoing, but also I want to give you the idea here. So mcDIV is calculated from the top row. But how do we get a reasonable node distribution for mcDIV? So Pan had this idea that we can permute the genes within each cell. We do the within cell permutation. So if we do it this way, then every cell keeps the same library size as the original cells. But here the genes are permuted genes. They are not the real genes. So therefore, we need to give this matrix its own negative control. That is, given the permuted genes, we perform within gene permutation again as in the top. Then these two matrices will give me the same statistic, but under the node. So the idea is that for the bottom row, we are getting it from the case where the genes are indeed uncorrelated. But the key thing why we need a double permutation is because for each matrix, we need to keep its genes, but disrupt their correlations. That's what we did in the top row, right? Within gene permutation preserves every gene's marginal distribution, removes gene-gene correlations. It removes cell library sizes. While when we do within cell permutation, we preserve the cell library sizes, but we remove the gene's marginal distributions. So these genes, the permuted genes, are no longer the original genes. So that's why they need their own negative control as here we have here. So that's the double permutation strategy. And I can show you that it worked. So this is a simulation in which we know the ground truth. So these are the metacells that are not pure. So they are composed of different cell types. And these are the metacells that are pure. So this is our statistic, mcDIV. So you can see that we are able to separate the two groups using the statistic. But how do we determine a threshold using permutation? If you just do within gene permutation, that is, you just do the top row without this within cell permutation, that's the problem. You are being too, too aggressive, or I should say too conservative. So basically you will call too many cells, metacells, as dubious because the threshold is just too low. But with our double permutation, by preserving cell library sizes, we get a much more reasonable threshold here. And we can also see that here, this left one shows how many dubious metacells we detected using double permutation. Not so many, but if we use within gene permutation, half of the metacells are dubious, which is not possible. And this also shows that using our double permutation approach, there's a much better separation between the pure metacells, the trustworthy ones, and the dubious ones than we use just within gene permutation. Due to time constraint, I'll just quickly show you two real data applications to demonstrate how this method we propose, mcRigor, can be helpful. The first one is to help with gene co-expression analysis. So this is a real data application from this Nature Medicine paper, and it was first implemented using the metacell method paper. So we reproduced the result. So the left column shows the gene-gene correlation matrix you get from single cells. Okay, so it's from patient data, healthy patient or COVID-19 patient. We hope to see some gene co-expression differences between the two groups of patients. But you can see that with single cell data, the correlations are very sparse because data are sparse. And if we use all metacells, and this is from the original paper, we can see that for this group of genes, block of genes, so these genes are basically adaptive immune response genes. We see some strong correlations in both groups of patients, both healthy and COVID-19. But using our selected trustworthy metacells, using mcRigor, we can see some big difference. So the same group of genes have weak correlation in healthy or weak co-expression in healthy patients, but strong co-expression in COVID-19 patients. This is consistent with the nature of these genes being adaptive immune response. They should be active in COVID patients, but not healthy patients. And looking at the data, we can also see why we have those fake positive correlations using all metacells. So here, the blue metacells are the questionable, dubious ones. The red are the trustworthy ones. So you can see this blue one here, drove the correlation to be very high, but they should be removed. And if you look at the single cell data, original data, you don't see such strong correlation there. So that's one example. The second example is for single cell multi-ohm data. We have RNA-seq and ATAC-seq. So basically, we're trying to draw some associations between enhancers and gene expression for inferring gene regulation. So we can actually see that using our trustworthy metacell versus using all metacells, we see some differences. And this is the data analysis from the C-cell paper at Nature Biotech. So we see that there's one edge used to be found by all metacells that disappeared. So if we look at the raw single cell data by cell type, we do not see a peak here. So which suggests that this association was indeed spurious. But if we look at this additional one we found using just trustworthy metacells, but missed by the previous analysis, we see that this is indeed a small peak in all the cell types here. By aggregating cells into metacells, we can discover this. But if you don't use just trustworthy metacells, by using all metacells, you will miss this one. So that's just two of the many examples we hope to show in this work. So in summary, I talk about three ways of doing permutation. The first one is the simplest one. For the DE analysis, we just permute the labels so we can get a negative case. The second one within gene permutation is for us to get some permuted cells whose cell-cell relationships are disrupted. And here we don't need to preserve cell library sizes because in this visualization thing, our score is defined as Pearson correlation whose range from -1 to +1. So we have the same range that's comparable between the cells and permuted cells. And for the third one, is the most tricky one because we had to do this double permutation to preserve cell library sizes when we need a negative control. So I just want to say that even the permutation may seem as an easy technique, but when you want to implement it in your analysis, it's very important to think what you want to preserve, what you want to disrupt, and then design the permutation accordingly. So finally, I want to thank my collaborators and my students and postdocs for the three projects. In particular, the first project, which is a surprise finding, has actually led to many how to say, feedback and interesting discussion about how to do differential expression analysis, an analysis of 10 years old. And for the visualization, we hope that we can help people visualize or have a better, more reliable t-SNE and UMAP plot by optimizing the hyperparameter. Finally, for the mcRigor, we're hoping to submit the preprint very soon, so you can see the whole idea there. Thank you very much.
Yeah. Hi, Dr. Lee. Thank you for your inspiring talk. So, because I see some paper doing like a perturbation-based method, like introduce noises to the data and to see whether, for example, differential gene remain the same. So, what's real different of the core idea between this one and the permutation one?
So you said other paper use permutation to introduce
No,
noise?
perturbation meaning
Perturbation.
Yeah. For example, adding some noises to the data you have.
Good question. Actually, that's related to another project in my group. I would say if you have heard about this bias variance trade-off thing in statistics, that's it. So basically here, our permutation is to provide a negative control for removing the bias. And when you add noise to your data, you are evaluating the variance of a result, how robust or how stable your result is.
So the rigor is about, the bias.
Bias, yeah. So what I'm introducing here is all about the bias. We're getting a negative control case. But I totally agree. I think variance is another thing to consider. Yeah, thank you.
It's very interesting talk, and I'm curious for the BOC-DE differential expression analysis one. Your observation is for some data sets the negative binomial distribution doesn't hold, but currently on single cell it's like Poisson and negative binomial distribution on their main distributions. I wonder, have you looked at them and how good are they, and how does that I don't know just affect? I mean, general.
Yeah, very good question. I would say that for single cell data, we have abundant evidence. We have a genome biology paper called Statistics or Biology, the Zero Inflation Controversy. It's a review paper. So in that paper, we actually found, and I think Xiang at Michigan here, he also had another paper showing that negative binomial is a good distribution if your cells are homogeneous in one cell type. So within that case, you can safely say, and also the single cell technology has the UMI, unique molecular identifier for removing PCR bias. So for that case, negative binomial is reasonable, which is also related to that two-layer model, right? The measurement model can be reasonably assumed as Poisson. And if the expression model is gamma, the negative binomial would be good. And because gamma is a very flexible distribution for describing positive real values. It has two parameters. Anyway, so I think for the question we report here for both RNA-seq examples, I think here just the patients are very heterogeneous. And they may have different genetic backgrounds, right? So when you pull them together, you don't use other patient covariates, just simply do the two-condition DE test, you might have false positives. But in here, we can discover that negative binomial doesn't fit well because of the large sample sizes, 51, 58. But previously, if you only have 3 values, there's no way to check. You just have to assume negative binomial. I think that's another difference. But for single cell data, I wouldn't be too worried because we usually have at least maybe 100 cells to look at, right? So we have a large number to check the distribution. Yeah.
Any other questions?
Thank you.
Hi, I'm very curious about the time complexity of your methods. But usually permutation-based methods.
That's a good question. I would like to say that the good thing about all the permutation I introduced here is that, except the first one, where we did 1,000 permutation, just to show the distribution of the DE gene number. But for the second and the third one, we only permuted once. Yeah, the reason is that we actually, after the permutation, every permuted cell will give me one null value, and they together give me a distribution. So I don't have to repeat this again and again. And the reason is because for both scDEED and mcRigor, we're looking at a per cell statistic. So we're not looking at the statistic for all cells. So that's why we don't need to get many, many rounds of permutation, which is a good thing to me. Yeah, so that's why it's not complicated. You're just doing permutation for once. Yeah.
Thank you, Dr. Lee.
No.
So my question is about like for the like some single cell RNA data analysis. If we want to focus on like some downstream tasks, like RNA velocity, something like this, how important do you think we need to do the neighboring embedding trustworthy check?
Definitely. That is something we are definitely interested in. Yeah. And I would say that's why I feel like probably this talk can inspire you to think about how it can be covered in your analysis, right? Some negative control where you know that nothing should be found as a sanity check. To see, because I think for single cell data analysis, the reality is that it has a lot of steps involved, right? We are not just doing a one-step analysis. So we need to be sure that some signals we found are not due to the previous steps, but should be really be in the data. Yeah, so I think Professor Jingyi, when he introduced me, he mentioned that I'm also advocating the synthetic null data idea to be added to data analysis. And I would say permutation is just one approach to implement the idea. Yeah. Thank you.
Do we have online questions?
Online question.
Okay, Jessica, I'll ask a couple. Still have a few minutes.
Yeah.
So for the metacell, you mentioned the hyperparameter.
Yeah.
Have you thought about making it different depending on whether it's a dense area or the sparse area?
Very good question. I have to say that, yes. So right now, for this work alone, we're just proposing a criterion, and we're trying to optimize each of the four methods in terms of their hyperparameter. And then we can compare these four methods to see which one is better for this data set. There are some results I'm not showing, but I would say ultimately we are devising our own algorithm. So when we devise an algorithm for defining metacell, I think it should consider density. So maybe the neighborhood size shouldn't be the same thing. So this gamma is just a way to summarize existing methods hyperparameter. Maybe it's not directly gamma, but basically have another parameter that controls the gamma. We're just using it for simplicity. But how do we divide single cells into trustworthy metacells by optimizing our criterion? That is something we're working separately as a new metacell algorithm. Yeah. Thank you.
Thank you for your nice talk. I'm just curious whether the SD is applicable to even image-based spatial transcriptomics, which is also single cell resolution.
Yeah, for sure. Because scDEED is just a visualization thing, right? It's an add-on. It's an add-on to t-SNE or UMAP. As long as you can use t-SNE or UMAP to visualize your data, you can try scDEED.
Great.
Yeah, thank you.
I'll load another one.
Yeah.
So on your second part about embedding credible versus dubious embedding, there is a parameter 50% of the cells.
Yeah.
Yes.
If you dial that down to 20%, would you get more of the rare clusters to be credible?
Good question. Yes, in this paper, we did try varying that neighborhood percentage, right? From 50 down to like 10% or 20% and up to 80%, 90%. Exactly, because t-SNE and UMAP are designed to preserve local neighborhood. If this cell neighborhood size is small, then you will find more cells to be trustworthy. Yeah, more embedding to be trustworthy because that's what it's designed for. But we chose this 50% because we want to talk about mid-range relationship, like here. Oh, sorry, I should go back. Where is it? Yeah, so I should go here. So basically we want to say whether the relationship between these clusters are reliable. So that's why we go larger, 50%. But the good thing is that we, in our, stability analysis or sensitivity analysis, we found that if you change the percentage to 40% to 60%, it doesn't change that much. Yeah, so that's what we did. Yeah.
Last one. Okay, I'll ask the last one. So in the final part,
Yeah.
there is the assumption that library size is purely technical.
Yeah.
And then when the size is bigger or smaller, the features are uncorrelated.
Yeah.
But biologically, I think it's a profound question on whether different cell types genuinely have the original size been different and something propagated into library
Exactly.
size.
Yeah, that's a very good question. I think here we are in a simpler situation because we hope that the metacell definition should just include cells of the same type, right? Every metacell, if it's trustworthy, it should only include cells in the same type. So that's why the cell type differences shouldn't be biological. That's what we hope. Yeah. Thank you. Yeah. Thank you.